Thank you so much for the invitation. It's a great pleasure to be here today with you and learning more about your organization and getting to meet you now. So as you said, today we're going to talk about how the brain makes language possible. And usually when we think about the brain, this is more or less an image that we have in mind. But for the argument of today, we actually want to dive deeper into the brain and look at what's going on underneath the surface, so all the connections in the brain and how they contribute. Now, when we think about language connections or connections in the brain and language, then really the first model that we have available is the original model from Wernicke, Karl Wernicke, that you can see here on the left side. And he postulated that you have a center in the frontal lobe for articulation, you have a center in the temporal lobe for comprehension, and those two have to be connected. And at the time, he called it the psychic arc. That is the connection between the two. And it was hypothesized to go through the insula. So you can see the dashed lines here is a very direct connection. Now, as time went on, the narrative changed. And instead of having a psychic arc through the insula, the models shifted towards a much larger tract called the arcuate fasciculus that goes through the parietal lobe and around the sylvian fissure. And that's what you see on the right side. So now you're connecting the center for articulation, also better known as Broca's area in the frontal lobe, with the center for comprehension, Wernicke's area in the temporal lobe. And you go via what's indicated here by A, which is the angular gyrus and the supramarginal gyrus. So together they form the inferior parietal lobe, and that part of the brain has been nicknamed, so to speak, as Geschwin's territory. Now, if we look at that in more detail, this is one of the original brains that Broca built his theory on, or two brains actually here, that you can see where in the inferior frontal gyrus, you see there is a lesion, and these brains were preserved. And what he's done is he recorded the clinical presentation of those patients while they were still alive, and then looked at the brain and put effectively one and one together and said these patients could not articulate, they were non-fluent or agrammatic, and there's a lesion in this part of the brain. Now, this theory is known as localizationism, where one part of the brain has one function. Now, this is what textbooks usually say, but I want to give you a couple more examples why we think the story is actually slightly more complicated than just this part of the brain does this function. So the first one is that when you look at a database of functional imaging, so in this case neuroscience, where all the functional data is stored, and you just look for the term Broca, you can see that we have a hotspot in what is classically considered to be Broca's area, but you can see that is definitely not an exclusive activation of that part of the brain. Also, fun fact, if you ask experts in the field where Broca's area is, we tend to not agree. So there's about a 50% overlap at max where Broca's area is. And for Wernicke, that is even more distributed. Okay, but let me show you some clinical data. So the assumption here is if you have a lesion to more or less Broca's area, then you would present with a Broca's aphasia, which means you understand language, so the comprehension is intact, but the articulation is non-fluent. Now, when we look at stroke data, there's increasing evidence over time where we can see that a chronic Broca's aphasia does not necessarily overlap with the traditionally defined Broca's area. And that challenges this conventional understanding of the role of Broca's area in chronic Broca's aphasia. So what you see in the images here is that the hotspots, so the hotter colors in this case, so going towards the red, are not in the classical Broca's area where you would then expect to be, but they tend to be shifted more into the white matter. Now, this is for stroke patients. We see a similar pattern in brain tumor patients. So here are several patients who were operated on for brain tumors. And you see a very similar pattern where a chronic Broca's aphasia does not typically arise from a resection of Broca's area, so taking that part of the brain out. But it occurs when other parts of the brain are damaged, most prominently the ventral sensory motor cortex and the supramarginal gyrus. So this challenges the classical localizationist idea of Broca's area being the driver for Broca's aphasia and the seat for language articulation. Now, when we go back to the original case from the original brain that Broca described, Nina Dronkers had the possibility to actually put the very same brain in an MRI scanner many years later. And she published that in Brain in 2007. And this is what you can see here, where you can appreciate that on the MRI, the extent of the lesion, the extent of the damage, goes far beyond Broca's area, deep into the white matter. Now, this was previously not seen because there was no scan of Broca's patient, but luckily he preserved the brain, so the study was feasible. Now, a couple of years later, my colleague Michel Thibault-Deschotten used this data and he reconstructed the lesion of Broca's patient here and put that on an atlas to see which white matter tracts would be affected by a lesion to Broca's area. And what you can see here is that the lesion that was originally described for Le Bon, or better known as patient 10, disconnected the inferior frontal gyrus from the parietal lobe, from the temporal lobe, but most importantly also from itself. So you had an impact on long-range connections and also shorter-range connections within the frontal lobe. So what these studies show us that the story has always been way more complicated than we appreciated, but we now have tools to actually look at that. So how can we look at the white matter? Slight detour here. So one of the methods that has been available for decades at this point is so-called Klingler postmortem dissection, where a brain is extracted and then goes through a process of freezing and thawing the brain multiple times. And that makes the gray matter quite brittle, so it can be carved away, and it exposes the white matter underneath. Now, obviously, this technique is quite demanding in terms of time, expertise, and specimens. So very luckily, about 40 years ago, a new method became available called diffusion-weighted imaging tractography. And that method now lets us study the white matter of the living human brain. And that means you can repeat the scans. You can also now do longitudinal studies to look at the changes in the white matter across the lifespan, for example. And you can also do studies where you compare more and more participants because it's quite easy to acquire the data with an MRI scanner. Now, some basic ideas about how tractography works. So I recently was invited to Vancouver, and as you do, you check where you are, you check where you want to go, and I found the optimal way to get there, in the case a flight, and it was a direct connection. Now, once you're there, you look at how to get from the airport to downtown, and once you're in downtown, I run on coffee 24/7, so I was looking for the next coffee place. And you can see that now I had so many different ways I could take to find coffee. Now, why am I showing you this? I'm showing you this because that directly relates back to how tractography works. So if we have really good data, we can see a lot more of the anatomy. If we have clinical data, we typically have less good data. And that means that depending on what type of data set you're working with, you can either look at the direct connections or you can really zoom in and look at the small interim connections in the brain. Now, tractography is also quite a good tool because we can not only use it in healthy brain, but we can also use it in pathological brain. So when things go wrong. So to stick with this example, that was the flight I had booked, but then that flight got cancelled. And when it rains, it pours. So my next flight also got cancelled. And instead of flying to Vancouver, I found myself going to Iceland. Now, that is akin to what can happen in the brain. Direct connections can be damaged and the brain finds alternative ways. But just as in this example, if we calculate the energy cost for this change, you can see in this case, it was a 1.5% higher CO2 emission. The cost effectively was higher to take the other route that is not the direct connection. And also in terms of time, we're not even going to spend any time discussing how long it took in the end, but you can see that when you lose the direct connection, things tend to take longer, and that is also what we see in the brain. So if we bring it back, this is again an example from stroke where patients suffered a lesion to the left hemisphere, and then they were scanned at various time points thereafter. And what you can see is that the peak activation in the left hemisphere reduced, and you see this activation emerge on the right hemisphere. Now, patients who could do the shift were better at recovering their language function, but the ones who recovered the best long-term were the ones who were able to reactivate the language network in the left hemisphere. So you can see that the brain has some potential to shift and dynamically adapt to changes in its environment, but you can also see that if the activation for language can shift to the right side, then this whole narrative of language is a left hemisphere function is probably also slightly underrepresenting the complexity in the anatomy and in our functional expression. So when we look at the anatomy of the language network, there is about 7 pathways in the brain that have been directly or indirectly linked to various language functions. And when we purely look at the anatomy of them, we can see that they're all present in the left and the right hemisphere. So they're considered bilateral connections. Now, Lilith Dolian in my lab spent her PhD on studying those connections in a lot of detail. And what you see in this scatter plot, in this plot here, is that she looked at a measure of the tracts that you can consider as the integrity of the tract. So that's the HMOA that you see. Then in the middle, we have TC, that's the tract count. So how many streamlines each tract had. And then at the right side, you have the VC, the volume of the tract. And she looked at each of these connections in the left hemisphere and in the right hemisphere, and then calculated the laterality index. And the assumption from the beginning would have been that if all these connections are exclusively language connections and language is in the left hemisphere, then you would expect these connections to be primarily shifted towards the left side, so be stronger in the left in terms of volume or in their integrity. Now, as you can see, that is not what she found, quite the opposite. So some of the tracts were left lateralized consistently across the 3 measures. Other tracts were consistently right lateralized. So if you look at the first line, the long segment of the arcuate fasciculus was left lateralized. But then in the second line, the anterior segment, so a frontal-parietal connection, was right lateralized, and other tracts were completely bilateral. There was no clear preference if they're left or right lateralized. She then did another analysis where she developed her own index to measure the variability in these tracts. So how variable is one tract in one hemisphere compared to the other? And again, there is no coherent pattern in terms of which tract or which system or network is most variable. So what that led us conclude is that the language connectome, and by language connectome we mean all these tracts taken together, is neither uniformly left lateralized nor is it anatomically fixed, but rather it is a probabilistic system where we have tract-specific asymmetries and variability patterns. And we're going to come back to that in a minute. But one of the exciting things that we also wanted to know is language is extremely dynamic across the lifespan. From our early words to our last words, we acquire a huge amount of vocabulary in the first couple of years, then we perfect our syntax, how to use those words in relation to each other and build up on the meaning and other things like prosody and understanding irony, for example, that comes later as well. Now, when we look at the developmental changes behaviorally, they're very prominent. And the question was, are these behavioral changes in any way mirrored in the anatomy of these connections? So what we did in this study that is just currently in press in human brain mapping is we took various data sets and combined them. And that gave us a longitudinal data set across the lifespan. So starting from birth all the way to the age of 100. So this is a gigantic data set in the end that we looked at. And again, we extracted measures or features, as it's called here, from the white matter, where you have things like the diameter, the volume, the tract count, and the integrity measured by fractional anisotropy, for example. And then based on that, you can calculate, again, the lateralization of each of these features of the white matter and go across all the different white matter tracts that we have in the brain. So when you do that for the arcuate fasciculus, the classical language connection, what we can see is that the arcuate overall tends to be left lateralized, and that's what we've already seen in Lilith's work, but it tends to be left lateralized across the entire lifespan when you look at measures of fractional anisotropy, so the integrity of the tract, but also the volume of the tract. Now, interestingly, if you look at some other indices like the mean diffusivity, the axial and radial diffusivity, or the length of the tract, you can see that it's not a consistent left pattern. So some of these measurements tend to be consistently stronger on the right, the length, for example, at the bottom right of the panel, and others are more susceptible to changes across the lifespan. So if you look at MD on the top right, for example, you can see that we start off slightly left lateralized and across the lifespan move to be bilateral, slightly right lateralized. And this is a pattern that we could observe across various different connections in the brain. Now, I started off with mentioning that there's this assumption that language is a left lateralized function. And you probably all heard at some point about the idea of being left or right brained. And what we were wondering is if it's not the tracts themselves that are left lateralized, is there maybe a pattern across them where if you have a stronger volume of the arcuate in the left hemisphere, that goes with, for example, a stronger volume of the inferior frontal occipital fasciculus in the right hemisphere, and to see if there is some kind of clustering that we could find across the language networks. So Lilith, again, did the study where she looked at all these 7 tracts in the left and the right hemisphere and was trying to see if there is a coherent pattern in language lateralization. And quite to our surprise, let me know if you see any pattern in this cloud of data points, but we did not find any coherent patterns in there, was that we don't seem to have clear phenotypes where you are more lateralized to the left or more lateralized to the right. But there seems to be a consistent gradient in terms of the anatomical configuration that we can observe across the population. Now, this also to some degree correlated with behavioral measures that we could see, but to a much lesser extent than we would have expected. Okay, so now we know that we're all different. Our anatomy is a gradient. The question was, does it matter? So in this study, we did a meta-analysis of 326 studies where they looked at the variability in the anatomy and the variability in cognitive behavioral measures. And a couple of things we learned from the study was, number one, that most of what we know about the function of the white matter, we actually know from clinical studies. So nearly 50% of the studies were conducted in neurological and neurosurgical patients, nearly 30% in psychiatric patients, and then only about 25% of the studies were conducted in healthy participants. The other thing that we can see is that some of the tracts are superstars in the field, like the corticospinal tract, while other tracts are a lot less studied. If we go all the way down to the bottom of the list, you see, for example, the acumbofrontal pathway. Now that in part goes back to what I mentioned earlier, that the data quality determines what you can look at. The corticospinal tract is a very prominent tract. You can get that even if the data is not optimally, whereas the acumbofrontal pathway is a really small tract. So now you're on the far right where I was looking for coffee in Vancouver, and you need much more higher resolution data to look at that. Now, the other thing that quite surprised us, if we zoom into the arcuate fasciculus, is that the arcuate was always considered to be the language pathway. But actually, when you look at the correlations with cognition, you can see that it has a strong correlation with language, that's correct, but it is not exclusive. You have correlations with other cognitive functions as well. And that is a pattern that we saw across all the white matter connections that we looked at. So an important point to take away here is that there is no one tract, one function association, but that these tracts seem to be dynamically recruited for different cognitive functions as they are needed. So a quick interim summary. We started in the 19th century with the classical model of Broca-Wernicke connected by the arcuate that goes behind the inferior parietal lobe. Then the early 2000s, we had the addition of this indirect segment of the arcuate where we have the anterior and the posterior segment that relay in the inferior parietal lobe or Geschwin's territory. And since then, many more tracts have been associated with language and linguistic functions through lesion studies, through TMS studies, through correlational studies, through direct cortical stimulation. Many methods have been used. And we're probably going to spend the rest of my career for sure trying to disentangle how this complex network actually works and what is needed for which function. Now, Peter Hagert and I recently tried to put the state of the art together in one diagram that looks like this, where every box is a part of the cortex, so one cortical area. Then in pink, we have the subcortical structures like the amygdala and the thalamus. And then the lines are all the white matter connections that we know that have been implicated to be relevant for language, and you can see them depicted in the top right as well. So you can see that we have come a long way from Broca and Wernicke 1 tract to our current understanding of language in the brain. Now there's one caveat that I want to highlight, and I just give you a second internally to think about who you might be seeing here. I'm not going to ask you, but these are all very famous women, and most of the time it takes a while for people to recognize who we're looking at. But in fact, these are all the women who won the Nobel Prize for physiology or medicine. Now, what you see in the graph below is the comparison of all the women who won it compared to all the men who won it. So it's a score of 26 to 600, except that it's recently changed slightly in our favor. Now, I asked one of my students, Eva Guzman-Chacon, to make an image of what you need to look like as a woman to win the Nobel Prize. And here it is. So this is the average face of a female Nobel laureate. Now, what you can see is that the algorithm worked. We have a face with all the right parts in the right places. But what you can also appreciate is that the image in the middle does not look like any of the women around it, and vice versa. None of the women around look like the image in the middle. And that's very easy to appreciate when you look at faces. It's a lot harder to see that when you look at brain imaging. So here's one example. You see the image I used earlier with a beautiful arcuate fasciculus, textbook arcuate. But in real life, when you actually look at individual dissections, what you can see here on the slide is that you actually have 3 participants. So that's the columns, the 3 participants. And all of them were scanned in different centers. So we have London, Cambridge, and Oxford here. And what you can see is that the variability between the scans, so between the sites, there is some variability, but it's not as huge as the variability between each and every participant. And that's an important thing to remember. There is a huge degree of variability between us. And most of the time in neuroimaging, we look at average reconstructions of brain anatomy. Now, this is obviously just an example of individual reconstructions of one tract, but we did this on a larger scale and looked at all the white matter in the brain in this paper. And what you see here is that there is a gradient of variability. So the parts of the brain that are older and deeper inside the brain tend to be more alike between us. And as we go more lateral, you can see in the warmer colors the areas of regions that are more variable between us. Now, that is extremely important to be aware of, because what we tend to do in neuroimaging quite a bit is we use an atlas. Now, an atlas works well in areas where we are alike. It does not work as well in areas where we know there is a huge degree of variability between us. And most importantly, when we bring that back to the patient, what we can see is that an atlas could potentially miss an entire tract entirely. So what you see here in green is a brain tumor, and that brain tumor had a mass effect. So it pushed the white matter slightly to the side. And we had diffusion-weighted imaging for that particular patient. So we reconstructed the corticospinal tract, one of the prominent big tracts that connects the motor cortex to the periphery. And what you can see is that tractography was able to reconstruct the tract beautifully. But when we asked an atlas to describe where that tract is, you see the red outline, the atlas completely and entirely missed where that tract actually was located in the patient's brain. So that is just something to be aware of when using atlases in neuroimaging. Now, the other thing that we were interested in is that obviously this is a snapshot of variability in the brain around the age of somewhere between 20 and 40, because this is based on a huge database called the Human Connectome Project. But the question was, how does this variability develop and change across the lifespan? So that's a study that we recently done in this large data set that I told you about earlier, where we were able to replicate the same study, but now across the lifespan at various time points. And you can see that even without knowing in detail what you're looking at here, that the images change over time, and that can also be measured, and that's in the table below, where we see that some tracts tend to change more than others. Now, we already knew that variability is related to variance or differences in cognition and behavior. So also in this case, we were interested to understand how that relates to various cognitive functions. And in this case, I'm showing you working memory, episodic memory, and reading skills. And you can see that the changes across the lifespan and the variability that we can measure in the data varies across the lifespan, and it correlates with various cognitive functions, sometimes at different times across the lifespan. And that's what you see also on the right side of the panel, where the data clouds are separated into development, so the early years, then young adults and aging in the later years. And you could see that the correlation lines vary between people. Now we know that we are variable. We know there is correlations with cognition and behavior, but the question that we also had is, can we use this information in a clinically meaningful way and potentially predict recovery when things have gone wrong? So one of the things that we did here is we recruited patients who suffered a stroke, so a lesion to the left side of the brain, and they developed an aphasia. And that could be any type of aphasia. We did not select them for comprehension or production deficits. And what we then did is we followed these patients over time, and we repeated the scan, and we repeated the language assessment. So what you see on the y-axis here is the language recovery 6 months after the stroke. And on the x-axis, you see the size of the arcuate fasciculus, but in the healthy right hemisphere. So they had a lesion to the left, and we looked at the arcuate in the other side of the brain. And when I highlight 3 patients here, you can see patient number 1 is somewhere in the middle on the graph in the left. And you can see that that patient did not recover language that well. And when we look at the arcuate in the right hemisphere, it's rather thin. Now, as we go through the examples of the patients, you can see that patient number 3 has a really thick and strong connection in the right hemisphere. And when we look where that patient is on the scatter plot, you can see that patient number 3 is actually in the blue box. And the blue box means that according to the neuropsychological assessment, that patient has recovered their language function back to normal, so back to healthy language. Now, what is interesting here is that when you look at demographic data or clinical data, you can predict somewhere between 30 to 40% of recovery by simply adding the size of the arcuate fasciculus, so the variability in that anatomy, into the model. You can now nearly double that prediction. So that tells us that looking at the variability in anatomy is tremendously useful when it comes to predicting clinical outcomes that we previously were not able to predict that well. Now, this was obviously a small-scale study done in about 20 patients, and then recently Leah Talozzi did this huge work where she looked at 1,333 stroke patients and took their lesion profile, put it into our software called the disconnector, and calculated the patterns of disconnection. So for every lesion, you get a map of all the white matter that is affected by that lesion. And she did that systematically for all 1,300 and then run the data of those disconnection maps through a dimension reduction tool called UMAP. And what you see in this chart is that patients who had similar lesions and similar patterns of disconnection clustered together. So the top left, for example, is all the right occipital lesions. And they cluster far away from patients that had different patterns of white matter disconnections. So that would be, for example, the temporal left on the bottom right. Okay. Now, the beauty of this approach was that she could then take another data set from Maurizio Corbetta, where they had cognitive and behavioral measures across 87 tasks, I think it was, where the patients were tested in the acute stage and again at 2 time points afterwards. And by combining those two data sets, what she could do is create what is now called the disconnectome symptom discoverer, where you have this effectively a map of the patterns of disconnection and the associated behavioral scores. Here you see the motor score, the language score, visual spatial attention. And what this allows you to do is you can take any new patient, take their lesion, bring that into that space and see where that patient lands. And that allows you to predict the outcome for any given patient 1 year after the stroke. So this is now a clinically useful and quite powerful tool. Now, trying to bring it all together, where does that leave us? So Michelle Thibaudet-Schotten and I, a couple of years ago now, did a review paper for Science where we looked at the different models that we have of language in the brain, starting with the classical model of localizationism, where you have Broca's area, Wernicke's area, and Geschwin's area in this case. And each area has one function. So Broca for articulation, Wernicke for comprehension, and Geschwin more for the integration. Now, when you then look at how much flexibility the system allows you to have by coding it where either the area is activated or not activated, you can appreciate that having just 3 areas and 2 brain states, so involved or not involved, doesn't really give rise to the complexity of the language system as we have it. So we dynamically interact in noisy surroundings oftentimes that would not be able to be computed in a simplistic model like that. So then we did the same with the so-called hierarchical model, where you have the same regions, but now they're connected by the arcuate fasciculus, for example. And the example here would be if you're asked to repeat a sentence, you would first get the auditory input, you would understand the auditory input, and then you would have to send that information towards the frontal cortex to articulate. Again, this model does not give you enough flexibility to really compute all those linguistic facets that we use in the time that we use. So what we proposed in this review is that language is an integrative model where areas of the brain and networks get recruited in a dynamic fashion and as they are needed. Now, one way to think about that is that if you ask me where language is in the brain, it's a bit like asking an orchestra where their music is. It's not in the single notes, it's not in the conductor, it's not in the instruments. It's in all of them coming together in a timely, dynamic fashion that music arises. But you need all the individual parts for it to work. And that is how we currently conceptualize language in the brain in my lab. So to conclude, I hope I could show you that Broca's lesion was never a purely cortical lesion, that the explanatory power of localizationism is not sufficient to explain language in the brain, but that language most likely emerges through a distributed white matter system in the brain, and that there is a structural variability gradient in the brain that we yet have to understand better, but that these individual differences that we can see also give rise to the variation in how this anatomical scaffold is built and used across the lifespan. So to conclude, the language connectome is a conserved structure architecturally, but it is flexible in its expression depending on what it is needed for or if it needs to recover. And with that, I look forward to your questions.